Okay, good morning, everyone. Thank you for dialing into the webinar this morning. My name is Birgit Meldal. I'm a curator here at IntAct, and I've got with me Piff, who is recording this session because it will also go onto the EBI Train Online website. And I've got Livia Perfetto with me, my colleague. She will be managing the chat. So if you have any questions, you can type into the chat box on the right-hand side, and she'll answer your questions. So let's get started. If you need to contact me later, and these slides will obviously be shared on the Train Online website, you can contact me by my email address or also via our Twitter handles. I'll be introducing protein-protein interaction, a little bit about the background. Then we'll be looking at the IntAct website as an example of a protein-protein interaction database, how to search, how to retrieve your data. We'll take a quick look at our integration services, PSICQUIC and IMEx, and hopefully we'll have a couple of minutes to introduce the complex portal. Okay, so what are protein-protein interactions and the networks? Protein-protein interactions are the physical and selective contacts that happen between pairs of proteins. And these have to be in certain molecular regions, like subcellular regions or extracellular areas, and in a defined biological context. So this could be in a certain cell cycle context. This could be a healthy versus disease state. This could be different manipulations of cell lines. And the interactome is the totality of all the PPIs that we observe, again, in a given context, like a cell, an organism, a cell line, manipulated cells, and so on. And to represent them, we can build this protein-protein interaction network, which is the graphical representation. And in here, all the dots or nodes are the proteins, and the lines between the dots represent the interactions. They're also called edges. Why do we look at the protein-protein interactions rather than the gene-gene interactions? So if you think about how a protein is produced, it's transcribed from DNA to RNA and then translated into a protein. But most proteins don't just have one unique function. They have lots of functions in different contexts within the cellular environment. Here's an example of two small networks for ARB2. So what can we derive from these networks? Firstly, we can predict the biological function of a protein that's so far been uncharacterized, because proteins that cluster together are assumed to have similar functions, and this is called guild by association. Secondly, to improve characterizations of protein complexes and pathways. So these interaction networks can work as a draft map that brings together all the detailed biological processes and pathways. As an example, here we've conducted an experiment in which cyclin T1 was used as a bait, and we've identified a number of preys. And one of these prey proteins is a new uncharacterized protein. But because we know a lot about all the other proteins, we can assume that this uncharacterized protein has also got something to do with transcription and cell cycle control. Similarly, we can infer from a network what a big protein complex might look like. For example, the human proteasome is difficult to isolate and characterize in its entirety. So people have identified small amounts of this complex and then built up the network and reconstructed the complex. Similarly, we can infer pathways from this network. So here we've overlaid the interleukin-3 function in the interleukin functions and identified the different interleukin pathways. Where does this data come from? So the data that we capture can come from any molecular experimental methods that capture protein-protein interaction, and they can be divided into two types. So the high throughput experiments are usually based on two hybrid assays, like the classic yeast-to hybrid assay, or mass spectrometry assays, usually as a second step after immune coimmune precipitations or pull downs. On the small scale or the low throughput ones, there are a huge number of different methods. One highlighted here is X-ray diffraction studies. What we need to remember is that not one single method can accurately reproduce the true binary interactions observed under physiological conditions, so unperturbed in vivo natural conditions, because every interaction detected experimentally is fundamentally artifactual. We always perturb the system in one way or another. We have to remember that what we observe could both be false positive as well as false negative. I've got a couple of slides about the properties of biological networks. Given a large network, about 80% of all proteins are connected in one giant cluster. People also call these the hairballs or the spaghetti monsters. These networks display clustering if the probability of a pair of nodes being adjacent is higher when the two nodes have a common neighbor. So that means if the two nodes are close together, or many of the nodes are close together with just one neighbor in between, then the network displays high clustering. The high clustering also infers that we have high modularity. That means that the functions that this network carries out are likely to be in a modular manner. And that allows us to infer pathways. Networks also show, or biological networks show, the small world effect. The median network distance is just 6 steps from one point to another. But something to really keep in mind is if interconnected binding networks could indiscriminately spread perturbations, so things that we knock out or don't exist in a certain biological context, how can individual pathways be turned on and off without upsetting the whole system? And this happens because these networks are also scale-free. In a scale-free network, the majority of the nodes have only a few connections, whereas some nodes, the hubs, are connected to many other nodes in the network. So you can see an example here on the right-hand side, and there are two different types of hubs. There's the party hub, which describes a network of connected proteins that occur in the same space and time, so at the same point in the cell cycle in the same subcellular location. And then we have some date hubs, proteins that connect these different party hubs, and these proteins occur in different subcellular, potentially in different subcellular locations and also at different times, and they may carry out different functions depending on the context. This makes these networks highly stable. If failures occur at random, the likelihood that a protein hub would be affected is small because the vast majority of proteins are those with a small degree of connectivity. So random failures might just happen in one of these small wagon wheel areas of a party hub. Even if a hub failure occurs in a network, it will generally not lose its connectiveness because they're the remaining hubs, and there's a redundancy within the network. However, if we lose a few major hubs from the network is turned into a set of rather isolated graphs. And that's what happens in cases of severe diseases like cancer, where we know that we usually need more than one mutation to upset the system and cause disease. But given the current limited coverage levels, because not all knowledge is already covered in protein-protein interaction databases, and also the quality of the interaction data, either based on the experimental background or the quality of the curation, the observed scale-free topology of existing interactomes cannot be extrapolated to the complete interactome. So when you reconstruct your personal network based on your data or the data that you've downloaded from a database, you always have to think about what is covered and what is the missing data. There are 4 types of protein-protein interactions. There are the direct binary interactions, which are the equivalent of a face-to-face conversation. We can measure these interactions by various molecular methods, such as like yeast to hybrids, calorific methods, or enzyme reactions, and many more. The main point here is that we know there are two proteins that are directly interacting. Often proteins interact in larger complexes. These are described as physical associations. These contain more than three or more participants. We compare these to a group discussion amongst people, and typical methods are pull downs or co-immunoprecipitations, but also things like crystallography methods, electron microscopy methods, and gel filtration assays. The third type of interactions are co-localizations or associations. These show two or more participants or proteins in closely related locations. We know they're very closely located at the same time, but there is no actual evidence that these proteins directly interact, because what we measure is an overlay of fluorescence in most cases. And these are the equivalents to a party where people might be there at the same time but never actually bump into each other. The last type of protein-protein interactions are the direct functional interactions. These are carried out almost always in vitro with purified proteins. This means we know exactly what we put into the assay. And examples are things like enzymatic assays, surface plasmon resonance, or crystallography. In IntAct, we don't just capture which proteins interact with each other, we also describe the binding regions where we have that knowledge from the experiment. And we can identify these with little blocks of interconnected regions, and you will see examples on the website shortly. We capture all sorts of extra information, and one thing we've added over the last year is a structured way of exporting all the mutation data. So if we have a data set where the mutation of one amino acid knocks out this interaction, then we capture this in very close detail. And here are three examples how we do this. We have a structured field that identifies the original sequence and the resulting sequence. I think my mouse can be seen. So here, this was a leucine to glutamine mutation at position 152 in this protein B2L11. We provide you with all the UniProt link outs, what this mutation does. So in this case, it decreases the interaction, and also the link to the publication and interaction. And we can do this for series of mutations. So for example, systematic alanine substitution assays, or also in cases where other amino acids are inserted, like this third example here. Okay, before we have a look at the website, one thing to consider is when we build networks, we have to extrapolate all the interaction evidences into binaries. A binary interactions means just A interacts with B. If we have more than two interactors in the assay, we have to make a decision how we reconstruct these binaries. And there are two methods. The matrix expansion assumes that all proteins interact with each other, which is okay for small-scale analyses, but imagine you have a pull-down assay and hundreds or even thousands of baits. A matrix expansion would become an enormous computational burden, and it would produce a large number of false positive results. So most databases, IntAct including, use the spoke expansion method where we assume one protein interacts with all the other members in the interaction. If the method had a bait, so a pull-down assay, an immunoprecipitation assay, two hybrid assay, then the bait becomes the center of the spoke. Otherwise, it is chosen by a random algorithm. Neither case is likely to represent a true reality, shown here on the right-hand side. as an example. But because we don't know the reality before we make the carry out the experimental analysis, we have to reconstruct it one way or another with computational methods. And you have to keep that in mind when you do your analyses, that anything with any interaction evidences that contain 3 or more proteins will introduce a certain level of false positive interactions and will miss some true positive ones. Okay. So IntAct is a publicly available repository of molecular interactions. We've now got over 800,000 binary interactions. These include the bespoke expanded inferred binaries. We've curated over 20,000 publications, and we have over 100,000 unique interactors in the database. These are proteins, small molecules, and also different types of nucleic acids which interact with proteins. And we have nearly 2,500 biological complexes in the database. All the data is standards compliant. So this is the PSI standard, which I'll come to later, and it is freely available via our website, the FTP site, or via the web service PSICQUIC. Again, I will explain more later. You are able to download everything and have a local installation. The way we curate data is by reading the papers or receiving direct submissions from our users. So please, here's a little plea. When you're producing your data and you're at the point of writing a manuscript and sending it to the journals, we would encourage you to deposit the data with one of the protein-protein interaction databases. The advantage is that we can release your data in parallel with your publication, and we can interact with you during the curation process to make sure we capture the data you produced absolutely correctly. And there's a link on our website to walk you through the submission process. Of course, your data will be held in highest confidence while your publication goes through review. When we curate a paper, we look at which experiments are provided in the paper. In this example, a yeast to hybrid and a co-immune precipitation. We then capture all the different interactions. So which proteins interact with which. And we go one step further. We don't just capture A interacts with B, but also anything we know about how these proteins were manipulated. Here in this yeast to hybrid example, we provide you exactly with which binding domain and activation domain was used. This is also the place where the mutations are captured, any experimental tags, and all the binding regions that we know of. Proteins are generally mapped to the UniProt canonical sequence, but if we know that an interaction is specific to an isoform, then we capture the isoform. And if we know that certain isoforms do not interact, we can capture that data as a negative interaction as well. We can also capture the post-process chains where the interaction is specific to the chain. We curate everything based on the manual and our standards. Everything in the database has got controlled vocabularies or CV terms. These are a dictionary that describes each term with a definition, and the terms are also captured in a hierarchy called the Protein Standards Initiative ontology. More later. Every entry in our database is checked by a second curator, and this process can take several rounds until both curators agree on how to capture the data correctly. Once the data is accepted, it will get released with our roughly monthly releases. And the data will go to the public website, the FTP site, and through our PSICQUIC services. And then some of our partner databases will again take this data and display at their front ends. We also export our interaction to other databases. Here is a flavor of some of these, but this is not exclusive. Okay, so how to perform a simple search. I won't do a live demo, but I've got some screenshots. You're welcome to go to the website, www.ebi.ac.uk/IntAct, and follow the process. I've chosen the example of CDK1, which is P06493. This is the UniProt identifier. When you search with this identifier, you get nearly 200 binary interactions. Bear in mind, if you repeat this exercise after the next release, these numbers might change. You can see that CDK1 interacts with cyclin B1 by various methods, but that table isn't very informative at first glance. So if you click on Customize View, it will open another column and another pop-up window. Click on Standard and Update, and then you'll get a more informative table. Okay, so in this table, you can see the interacting partners and the UniProt identifiers, as well as the species these proteins came from. And then a whole long list of interaction evidences by different methods. Everything is related, is cross-referenced to their PubMed ID. You can see what type of interaction this is, and interaction AC. You can see that IntAct captures not just data curated by IntAct curators, but a number of different partners. On the homepage, there's the whole list of about 12 partner databases, and they're all credited at the level of interaction. In the last column here, you can see whether this data was a binary experimental data, in which case it says NA, or whether it was expanded by spoke expansion. 15 interactions are visible per page, and you have to toggle through the page using the numbers above the table. You can look for other results at EBI databases for P06493 by clicking on the link, and then it opens up link outs to other resources. Okay, so obviously CDK1 interacts with more interactors than cyclin B1, and the quickest overview you can get is by clicking on the graph. This might take a little while to open up for you. So you can see all the unique interactors, mostly proteins, but there are quite a few small molecules as well, indicated with the triangles and their CABI full names. Clicking on any of these nodes highlights some properties on the right-hand side. And here I highlighted the CDK1 that's not part of the main wagon wheel. And you can see that this one is not the canonical human CDK1, but it's one of the isoforms, isoform 2. And if you click on the UniProt AC, it will take you to the UniProt entry. Here you are. Okay, if you want to know more about these interactors, you can go to the Interactors tab. You can click through the details. So again, you can toggle through all the different types of proteins, 107 different protein interactors, and you can go straight to the UniProt details for these UniProt entries. You can also do a second round of search by selecting one or more interactors from this list using those boxes on the left. Once you've selected these, click on Search Interactions, and then you can find a bigger network. So now we've got nearly 6,000 interactions for a list of CDK1 and its primary interactors. Again, you can see the list of interactors, and now we have all sorts of different types. We have nearly 2,000 proteins, but we also have 23 different compounds. And if you want to know more about these, you can go to the CABI link outs. We also have 8 different nucleic acids. These are mainly used as baits in things like EMSA supershift assays. And we have 3 genes that interact with some of the proteins in the network. And these are linked to the ensemble entries. If you go to the Graphs tab, this will take a while to load up if you're doing it live. This is the full graph of all the interactions that we've just searched. So this is nearly 6,000. It's not that easy to identify what's where, so you have to search a bit. You can zoom in and out. A little advertisement. We are currently working on a completely new technology to present you this graph, which will be much more interactive than the current Cytoscape web application we're using. So I've focused a bit more on what's oh no, sorry, that's later. So in this graph, you can now identify which interactors are interacting by, again, clicking on the nodes and seeing the details in the node properties. You can also manipulate the graph and use 3 different types of layouts. Okay, so let's try a different search. Instead of searching with the UniProt IC, we search with the actual gene name, CDK1. We now get a large number of interactions, about 3 times as many as we had before, because CDK1 is not specific to the human CDK1, but we get all sorts of different species. So here's the interaction network that you get from this search. And as you can see, there are also interactions for mouse CDK1, for the yeast, and for Drosophila CDK1. In yeast, for example, CDK1 is the synonym. You can see how our database captures terms from different fields, and all synonyms for gene names and protein names are captured. The little clusters on the bottom are highlighted here. So CDK1 also exists in other species, like a few other fungi, Candida albans, and Schizosaccharomyces pombe on the right-hand side. We also find some tremble entries for human and for mouse, because CDK1 is still the gene symbol for those entries. There's an isoform for human, and there's another fungus here, and the rat also is represented. So essentially, this is a warning. If you search with any kind of name, this is not species specific. Okay, so what else can we do? Of course, you want to download your results, and this you can do by clicking on the select download format. Oops. And you have several options here. You can download in a tab delimited file, which you can import into Excel, and is very nicely human readable. We provide the entire data for each interaction in our XML files. You can also export BioPACs and RDF formats, and if you want to do a full network analysis, you need to download the XGMML data type. If you're suspicious about the way n-array interactions, so those with 3 or more interactors, are expanded, you can filter them all out by clicking on the Filter Out button. As mentioned earlier, you can look up your proteins directly in UniProt. And now we have a look at the actual interaction details. You can either open them by clicking on this magnifying glass or the accession number. All interaction details start with the publication that it comes from and the type of experiment that was carried out. So in this example, it was a filter binding assay that was carried out in vitro with known predetermined participants. We also credit the partner databases who've curated this example. So MINT in this case. The interaction is direct because there are just 2 participants in this interaction, and it was an in vitro assay. We capture the figure legends where we know them. These might also be the tables. This can be from supplementary data as well as the primary publication. In the participants, we show you the experimental roles. So here we have a bait-prey relationship. This could also be unspecified or neutral. These little numbers on the right-hand side provide you with pop-up windows for further details. A stands for further annotations. S for stoichiometry. In this case, it's a one-to-one relationship, and which you can only see if you click on it, and experimental features. The experimental features are also highlighted on the bottom of the slide, because experimentally it was shown which binding regions in the protein were actually providing the interaction, and hovering your mouse over this orange bar will pop up with the details. These details can also be linked in the database, and then again hovering over provides you with the full detail. Okay, what else can we do? We can carry out advanced searches by clicking on Advanced Search and then show advanced fields. For example, we can search for all human interactions. And there are nearly 400,000 binary interactions that are linked to species human. But that might be a bit too much, so we can limit this. For example, if you click on the Advanced under the little search button on the top right corner, you can then specify your search further. You can see that species human is already captured, it's saved. And you can, for example, decide that you just want all the human crystals. You need to look up the term for this in the ontology lookup service. The link is at the end of the slides. We've now found just over 2,000 interactions that are specific to human with the method X-ray crystallography. However, we still have non-human proteins in this set of results, because human just specifies that any one of the proteins has to be human. So how do we find all human-human interactions? We have to use a slightly more involved search string. So we have to identify the tax ID for both A and B molecule. And then this reduces the number again. How do you find these details? If you go to the advanced search site again and click on MyQuery syntax reference, this opens up a pop-up window with a table with all the examples to search the different fields. You can make very involved and complicated search strings this way. And there are more search tips on the top. You can also do ontology search. Again, just underneath the search terms we just looked at, you have a text box. And when you start typing, it provides you with autocomplete suggestions. These terms come from the gene ontology, and they're linked to any annotations to the proteins at the protein level. You can also search by chemical structures. This widget is an import from the KEBI database. Okay, so how can we make more of protein-protein interaction databases? And this is what we've got PSICQUIC for. You might have noticed at the top of the table for any search results, there might be a list of link outs to other databases. So in an ideal world, we would have one central database for all protein-protein interactions with one user interface. However, a lot of these databases were set up independently 15, 20 years ago, and they weren't that easy to combine. But what was easier was to provide a common user interface. But we had to use standards and ontologies for this. There isn't much time in this webinar to talk about standards, except to highlighting that we use the Proteomics Standards Initiative for molecular interactions. And you can find all the details in the OLS. As I mentioned, the link is at the end of the slides. This initiative was started over 10 years ago, when it was clear that we have to work together with the different databases, because we were essentially curating independently, and lots of papers were curated many times in different databases. And after the standards were developed, we also decided that we should provide a non-redundant data set of all manually curated data. And that data set is captured by the International Molecular Exchange Consortium, or IMEx. Both the PSICQUIC and IMEx data sets provide a common format and schema, common controlled vocabularies, a common reporting guideline. There are two levels of information that we require. The IMEx is the absolute minimum level of information for each interaction that's required, and the IMEx level captures lots more in-depth information, including all the feature annotations. We provide a common scoring mechanism and a common data distribution, and that's the PSICQUIC service. PSICQUIC stands for Proteomics Standards Initiative Common Query Interface, and other than IntAct, PSICQUIC is a unique hit in Google. What does PSICQUIC do? PSICQUIC takes the user query. This can be a single gene name or protein accession number, or a complex MyQL query. It queries the registry for the location and availability of the partner databases before it goes to the databases and actually captures and retrieves all the relevant data. And then it provides both a tabular and a graphical output to the user. And this is what you get through the link out from the IntAct website. You can obviously search directly from the PSICQUIC website as well. And this is the example for all human X-ray crystallography interactions. The totality in PSICQUIC is over 25,000 interactions. And if you click on one example, like the complete IMEx compendium, again, you can see the table. Here, this is now slightly abbreviated compared to the IntAct details, but you have the same options of downloading the data and looking at the graphical representation. For small numbers of interactions, less than 5,000, you can directly cluster them. If you want to know more about the PSICQUIC registry, you can go to this link, and it provides you with all the details about the databases that are partners and how to search them with their REST and SOAP APIs. How do these databases differ? Well, here's an example. So the difference between the IntAct or the InterPorg database. The left-hand side screenshot is from the registry, and the right-hand side screenshot is from the PSICQUIC view website, where you can find all these tags by clicking on the magnifying glass. You can see that IntAct and the IMEx compendium contain not just protein-protein interactions, but also those with small molecules and nucleic acids. They're manually, internally curated, spoke expanded, and based on evidence. Whilst we also have lots of partners that run what we call data warehouses. They import data from the primary databases and combine it all. And InterPorg is such an example, and they're focused just on the protein-protein interactions. And they don't just import the evidence, interactions based on experimental evidence, but they also have a prediction algorithm. So you can choose what level of detail you like for your analysis. So how does IMEx differ from this? Here's the IMEx website. IMEx is the non-redundant set of these interactions, and they're all manually curated by us curators. IMEx is a subset of PSICQUIC, as you saw in the previous screenshot. And IMEx is slightly more recent than the PSICQUIC establishment. And the link to IMEx from any IntAct search is the second line of these link outs. And this is what the website looks like, again, when you search for all human X-ray crystallography evidences. You now just have one hit for the entire IMEx compendium, and you could cluster this query. Okay, what are the problems with this? As mentioned before, the curator interactions represent only a fraction of our current knowledge, let alone all the stuff that we haven't even experimentally identified yet. Different databases have different curation strategies. So IntAct curates absolutely everything, but we all obviously have focused topics that we work on at any given point in time. And other databases may be specific to a certain species or a certain type of interactions or certain molecular contexts. So the whole compendium of protein-protein interactions has got a big bias towards biologically interesting and well-funded topics. We're currently working to reduce this bias by looking at all these known but not yet curated interactions. Of course, text mining can increase the number of interactions in the databases, but that lacks the quality control that you get with the manual curated data. But through PSICQUIC, some of our partners do employ text mining, so you can capture that data if you like. Again, all types of experimental setups are inherently artificial, and they produce false positive interactions. So proteins that would never meet each other in vivo, because they either occupy different cellular compartments or occur at different times. So always keep in mind, if results in your network look strange, this might just be the artifact of the analysis or the experimental method. And of course, these databases curated by humans, so we may have made mistakes. If you find anything that looks weird, please get in touch and we'll have a look at it. How to get in touch? You can email us or contact us through the feedback button on our homepage. Here on the homepage, you also see all the partners that work with IntAct and use IntAct as their curation tool. You can sign up for a newsletter. We provide you with news 2 or 3 times a year. And you can follow us on Twitter. All of our releases, training events and attendance at conferences are tweeted through our Twitter account. This is the slide I referred to, so lots of useful links, including the ontology lookup service, third one from the bottom. That will come in very handy if you need to make more detailed searches, and the gene ontology is also very useful. You can find more tutorials on the Train Online website if you select the Systems filter on the left-hand side and then go to IntAct, and the IntAct tutorials as well as network analysis tutorials. If you're really interested in taking the network analysis further, I would suggest you do the 3-hour tutorial first. Okay. So we have time to have a quick look at the complex portal. Okay. So what's the difference between the curation in the complex portal and in interaction databases? So in interaction databases, all the evidence is based on individual experiments, whilst for the complexes, we often amalgamate the evidence from lots of experiments. We capture all of the evidence separately in IntAct, whilst we often have to bring evidence together from different publications as well as different experiments for the complex portal. Interactions evidences can come from experimental data or, as mentioned, through predictions or mining algorithms. This is very similar for the complexes, but we often have to go one step further, and using all the related data that we have, we might have to infer the actual evidence for the complex and its composition. And examples for databases are IMEx members for interactions and the complex portal for complexes. The complex portal, like all other databases, is freely accessible, is manually curated, and we believe of high quality, and we capture all macromolecular complexes. What is a complex? We consider a macromolecular complex as the biologically functional unit in the cell, rather than just the experimentally shown interaction evidence. These have to be at least 2 interacting molecules, and they have to be co-purified and be shown to have a functional function in the cell. We capture non-protein molecules, so small molecules and nucleic acids, as long as they're integral to the complex. What data do we capture? Every complex has a recommended name, is species specific as identified with the NCBI number, and has a unique complex accession. You can download the data or save it in the basket for further use. We provide you with what type of experimental evidence we have. So have we got full interaction evidence, or did we have to infer it based on background knowledge? And the little flask is either half full or empty, depending on the level, if it's not experimental evidence. We have a nice viewer which provides you with details of the stoichiometry and topology, and I'll come to it in a moment. The legend here describes what kind of molecules are in the complex, as well as the stoichiometry again in the table, and their role. So often we can identify the enzyme within a complex, or its cofactors or regulators. All complexes are annotated with a manually annotated function statement, which describes what it does, how and where. We also annotate the complexes to the gene ontology terms, and these are complex specific rather than protein specific. Where applicable, we capture the enzyme commission numbers, ligands, agonists, antagonists, or further comments. All lists are abbreviated to the first 5 members of the list, but you can expand it by clicking on Show All. We've integrated the Reactome widget for pathway details. If you click through the links on the top, either by complex identifier or pathway identifier, you can find the complex in the different biological contexts that it's been curated in Reactome. And you can go directly to Reactome website to find more details. Many of our complexes also have a free text property statement, which describes more about their molecular weight or binding properties or cofactors that need more explanation. We have a standard way of describing the assemblies, so whether it's a heterotetramer or homodimer and so on. And we've imported the Lightmole viewer from the PDB, and again, so here are the physical properties that we can capture, and we can link out directly to the PDBE instance. The last widget we implemented in the website is the gene expression viewer from the Expression Atlas, and again, you can find more details by linking out through their link outs. And then all complexes are also annotated with the cellular component in the gene ontology, and we've also started to capture the subcellular location separately. We annotate everything to diseases where applicable, and this is linked to the experimental factor ontology that's also held at the EBI and found in the OLS. And finally, drug targets are annotated via link to Chembl. We have other external resources like MatrixDB, and the cross-references can be found at the bottom of the page, as well as further reading. These are papers that either describe the function more or nice reviews about the complexes. And finally, all complexes are annotated with all their synonyms we can find, as well as the systematic name. The systematic name is the concatenation of the gene names together with the stoichiometry where applicable. Very quick look at the viewer. This is very interactive. You can click and drag these icons around. And if you click on it or click on expand all, you can zoom into the amino acid sequence. At first, it just opens a short bar. If you hold Shift and then click, you can zoom in down to the amino acid residue level. Hovering over the binding regions or the regions in the protein provides you with the details of the interaction binding sites. We also overlay information about the superfamilies, and you can collapse it all again when you're finished with the detailed view. You can highlight unique participants, up to about 20 different ones. And if there are more than 30 participants, then we collapse the stoichiometry to provide a better overview. You can export this by SVG. If you want to contact us, you can go to the website. So on the website, you can search in the search box. Again, this is not just for unique terms. You can use lists of protein identifiers as well, or make complex queries similar to the IntAct queries. You can also search by species and gene ontology terms. And there are 3 different filters at the first level of search results: for the species, for the biological role, and interactor type. Searching by organism provides you with an overview of the 20 organisms we have so far and how many complexes we have per organism. The basket provides you with the list of saved complexes, your personal list of saved complexes. The downloads option still to come. And the programmatic access provides you with the data in XML format. We still provide you the 2.5 format for interaction details, but we recommend that you move over to 3.0 because that captures the complex data in much more detail. We also provide a tab delimited format for import into Excel and a JSON that you can query through the web service and which the viewer also consumes. You can contact us through the big yellow box. It opens a web form where you can send comments as well as requests for complexes. If you found a bug and you have a GitHub account, please report it directly. Sign up to a newsletter. This is the same as the IntAct one, or follow us on Twitter. On the website, there's a quick tour and also a full in-depth webinar for the complex portal. So that is the end of my presentation and the link to upcoming webinars that the EBI provides. We would like you to fill in the survey that will launch at the end of this, at the end of the webinar. So thank you for your attention.